Hello and welcome to this lesson for Advanced Higher Biology. In this one we're going to be looking at movement through membranes. So previously we've been looking at membrane proteins and the difference between transmembrane and peripheral proteins and how they're associated with the phospholipid bilayer. Today we're going to have a look at the movement of molecules across that membrane before we move on to look at ion transport pumps. So what you need to know is understanding of how the proteins move molecules across the plasma membrane. You need to be able to describe the formation and the function of a channel protein. You need to explain how diffusion is controlled in both ligand-gated and voltage-gated channels. You need to describe the action of a transporter protein and explain how active transport allows molecules to move against a concentration gradient. So as we mentioned at the end of the last lesson, we have channel proteins which are transmembrane proteins spanning the entirety of the plasma membrane. Now the structure of these are multi-subunit proteins, and the subunits are arranged to form a water-filled pore forming across the membrane. So if we look at it from the side, you can see the 4 different subunits of this one, and from above you can see the 4 different subunits and the little pore being formed in the middle. And that's going to allow the facilitated diffusion of specific molecules. So each different channel protein will allow one specific molecule to move through. Now, some channel proteins require no conformational change, and that allows our facilitated diffusion as passive transport of a specific substance. There are other channel proteins, however, that are gated, and they will change their conformation to allow or prevent diffusion. So these can be either ligand or voltage gated. In the case of ligand-gated proteins, we have specific signal molecules which will bind to their corresponding receptor sites on specific proteins. So previously we've mentioned that specialised cells are all going to have specific proteins to require them to carry out the particular roles required by those cells. And this is one example of why. If this was a particular neurotransmitter or a hormone, we need a protein with an appropriate receptor site in order for that to be able to bind. So if you remember from previously, we talked about how a ligand is just something that can bind to a protein. In this case, we're looking at something specific. So the ligand in itself may be a neurotransmitter or hormone or another molecule that's required to bind to a specific receptor site. So in the first picture here, our protein is closed and isn't allowing any specific diffusion of any molecules through. When our ligand, our neurotransmitter here, has bound to its specific receptor site, however, that's going to change the conformation of the protein. So if you remember when we looked at the structure of proteins, we talked about how all of those amino acids making up that protein have their specific R groups, and it's the interactions between the different R groups that are causing the shape of the protein to be in the way that it is. When these ligands, whether it be a neurotransmitter or a hormone or any other type of signalling molecule, bind to their receptor sites, that's going to change the reactions between those R groups and cause a conformational change in the protein. As a result, in this case, it opens up and allows diffusion of the specific molecule that it requires to either come into the protein or to come out of our cell. So slightly more complicated diagram of the same thing. In this case, we have our phospholipid bilayer. We've still got our protein, and we've got our receptor sites for our neurotransmitter. And those of you who did higher human biology might remember from last year, we've got acetylcholine, in this case is our neurotransmitter. And we've got some ions on either side of the membrane which aren't able to travel across right now. Once our neurotransmitter binds to our receptor, that causes our conformational change and allows our ions to be able to get across our plasma membrane. So this is still diffusion, they're still moving from a high to a low concentration. So in this case, we've got our sodium, for example, is moving from a high concentration to a low concentration on the other side of the membrane. We also have our voltage-gated channel proteins. And in this case, we're referring to an ion concentration on either side of our plasma membrane, which is causing the protein to remain closed or to remain open. So in this case, if this is the outside of our cell and on the inside of our cell, it's more positive on the outside and more negative on the inside, and that's allowed it to be closed on this side. and open on the other side. If that situation is reversed, however, that causes our conformational change in the protein, and therefore our protein has ended up open and is allowing movement across it. It's not always the case, however, that needs to be reversal of positive to negative. If we look at this example, similarly, we've got our channel has been closed over here, our voltage is down at minus 70. However, all that we've required a little increase over here to minus 50, and as a result, our voltage-gated channel is now open, and that can allow some of our molecules to move across. So it may be that the presence of these particular ions are changing the concentration and are changing our voltage on one side, and therefore allowing it to move. So once we've got high concentration, that's changed our voltage and allowing them to be opened up and moved across our channel protein. There's another type of protein which is slightly different to a channel protein. In this case, we have a transporter protein, and these actually bind to the substance which needs to be transported. So channel proteins are a little bit different. If they're going to bind and have a conformational change, that change is to allow the free movement of their particular substance. A transport protein, however, will bind and cause a conformational change, but that's to directly move the specific substance across the membrane. So what's important about transporter proteins is that they are going to alternate between the two conformations. So for example, in this one here, it's open on this side. Our substance has approached it, has bound to its particular receptor site. That has caused our conformational change, so it's then become open on this side and released the substance into the cell. After that, this protein will just stay as it is. So it's going to stay with its open section on this side of the membrane before another molecule can come along, bind to it, and be transported out of the cell, where it will remain open for another one to come through. So in this way, they alternate between the two conformations, and that allows the binding site for the solute to be exposed on one side of the bilayer, then the other, and that happens in sequence. So again, just another example of a transporter protein. In this case, it's our glucose transporter. And we've got our phospholipid bilayer here and our protein and its particular receptor site. So our glucose has come in, that's caused a conformational change, and that's released our glucose molecule over here. Now this protein will stay like this, so it is now closed to the other glucose molecules that are around. They'll have to go off and find another glucose transporter protein to be used until this one can move another molecule of glucose back out of the cell, and then it will be open on that side. So that will remain closed until the same reaction can happen in reverse. But again, it's still diffusion. We're going from our high concentration of glucose to our low concentration of glucose. There are some transporter proteins which actually require energy in order to bring about this conformational change. And those are the ones which are used in active transport, i.e. they require a source of metabolic energy. So a good example of active transport is looking at the sodium-potassium exchange pump. So this is when we've got pump proteins that transfer substances across the membrane against their concentration gradient. So we're looking at molecules moving from a low concentration to a high concentration. Now, in this case, because we're moving from a high to a low concentration, we require some kind of energy, and that comes from the hydrolysis of ATP. So looking at this example, our sodium-potassium exchange pump here, we've got our sodium ion on this side of the plasma membrane and our potassium ions on the other side. Sodium ion is going to bind. We need to phosphorylate our protein, releasing phosphate from this ATP molecule in order to release the energy to cause the conformational change to allow these sodium ions to get across to the other side of our plasma membrane. So once we've had that occur, we've released our phosphate from our ATP and caused our conformational change. We've released our sodium ions, and this is why it's referred to as an exchange pump, because at the same time we have our potassium ions coming across to the other side of the pump. And then it's ready to start again. We've got more sodium ions waiting to come and bind along to here, and another ATP molecule coming along. So often we have pumps which are involved in active transport are actually transporter proteins coupled to an energy source. And some of those active transport proteins hydrolyze ATP directly in order to provide the energy for this conformational change required to move the substances across the membrane. So when we're looking at active transport, we're looking at a transporter protein. So it's going to have a conformational change after it's bound to the substance that it's transporting. But in order to gain the energy for that conformational change, it needs to hydrolyze some ATP to give it that energy and allow it to transport substances from a low concentration to a high concentration. So in summary then, we've looked at transport across membranes. We've looked at the simple diffusion of molecules such as oxygen and carbon dioxide, which are small non-polar molecules and are able just to make it across the phospholipid bilayer. We looked at facilitated diffusion through straightforward channel proteins, where our molecules can just move through the pore of whichever membrane that they're appropriate to. We've also looked at the fact that in channel proteins, some of these may not always have an open pore. They may be gated and require either the binding of ligands or a change in ion concentration to allow the gate to open and for the movement of that molecule to happen. We've got transporter proteins, which are going to be the ones which have a change of conformation, much like our gated channel proteins. However, in this case, conformational change is as a result of our molecule that we want to transport binding to the protein. So the molecule binds to the protein, we have a conformational change, and it's released. These are all examples of passive transport because we're going from a high concentration to a low concentration, so we don't require any energy. We do also have active transport, where we're moving from a low concentration to a high concentration, and this is going to require energy. Still a transporter protein, still having a conformational change and moving our molecules across the membrane, but needs energy in order for that conformational change to happen. So you should be able to describe the formation and the function of a channel protein, explain how we control diffusion in ligand-gated and voltage-gated channels, describe the action of a transporter protein, and explain how active transport allows molecules to move against our concentration gradients. As ever, please feel free to ask any questions you've got when we go over this in class.